We all like to eat and drink, whether it's a cold glass of beer, a nice milkshake, a juicy hamburger. All of us like to indulge in good food and drink and enjoy eating quite a bit. But what we don't always think about is we don't always think about what are the processes that happen behind the scenes to enable the food and drink get to us. What are the microbiology that happens that enables us to have that cold glass of beer that just has just the right flavor and the right amount of alcohol to make us enjoy ourselves? Or that nice sharp cheese which gives it that sharp tangy taste. Lots of microbiology goes on behind that. But on the other side, unfortunate microbiology goes behind it when you eat food and it makes you spend too much time in the restroom or the toilet, or the food, what makes your food go bad after it's been in a week in the fridge, and right before you want to take it out and eat it for lunch, you find out that there's a mold growing on it. So there's a lot of microbiology that happens and goes on behind the scenes with respect to our food, whether it's yeast, bacteria, or viruses. There are good actors and there are bad actors, some that promote flavors and make the food better, others that spoil the food or make it go sick. And in this series, Microbiology Explained, we're going to dive straight into this. Hello, my name is Dr. Dr. Ukpong, and I'm here to present to you on Microbiology Explained. And we're going to talk a lot about specifically food microbiology in this part of the series. We're going to delve into the detection methods for food microbiology, how we detect the good guys, how we detect the bad guys, what roles they play, etc., etc. And in the series of videos, we're going to talk about many of these topics of interest to you guys out there. First of all, for today's topic, though, we're going to start with a very, very simple detection system, which has been around in the food industry for a while, and it's very, very useful. We're going to talk about ATP, this molecule that you see on the screen right now. Don't worry about the science behind the molecule. As interesting as it is, what I really want you guys to take from this is that this is a very useful molecule, and it helps us in our environmental program. For those who don't remember, an environmental program is essentially how do you monitor the environment for bacteria, fungi, viruses, and stop that environment from hurting the food that you're producing. Or if you're in a hospital environment, hurting your patients who will encounter the surfaces which are there, whether it's the operating room or the scapula or whatever tools you're using. So before the ATP detection method was really developed back in the 90s, 80s, the way you would tell whether or not something is clean is essentially you go through the room, clean everything, and you look. You hope there's no bad smell, you hope it doesn't look bad, or if you're more sophisticated, you take some samples, you send those to the lab. 3-4 days later, those results come back, and there you know your room was cleaned 3 days ago. And so this wasn't really, really efficient. It wasn't quick enough. There wasn't good enough turnaround. And also sometimes it's expensive, and sometimes you just need the results much quicker. Hence, the ATP detection method was developed. There are many things to know about this molecule, but the chief thing I want you to know is it's present in all living cells. So every living thing has it, and recently dead things. So whether it's a piece of lettuce, or whether it's your hair, or some skin cells, or bacteria, they all have ATP in them. Second thing I want you to remember is that it's an energy molecule, meaning it has a high amount of energy in it, and cells use it for energy. And that will come into play later on in this video. So, how does an ATP detection system work? Well, it has two components. The first component is the bioluminometer, and then the second component is the reaction tube. The entire system works by trying to detect how many cells are on a surface. If you clean a surface, there should be less cells. If there's less cells, there should be less ATP. Remember, every cell has ATP. So if you clean the surface very well, you should reduce the amount of cells present. If you reduce the number of cells present, you will find less ATP on the surface. So these two components, the first component is the bioluminometer that you have here. This is from 3M. It's a really good machine. I use it a lot with my clients. It's wonderful. It works for me. But there are several good systems out there. It's your choice which one you choose to use. It's essentially a very, very sophisticated light detection system. So it detects light within the chamber in here, and it changes that right to some numerical reading over here that you see, 100 units, 200 units, etc., etc. That's the first component. The second component is this, what I call the reaction tube. Notice that there's a little bit of fluid in it right over there. In the reaction tube, there are two things that you should note. One is a detergent, almost a soapy thing, almost like the ones you use at home, but a little bit more sophisticated than that. And that breaks open the cells. And that breaking open of the cells allows whatever ATP is in those cells to come out. So usually when I go to an environment, I pull this out and I swab a certain area, a skin, hand, surface, whatever the case may be, depending on what I'm looking for. You take that and you push it in right here. Simply put, you put it into the swab. There you go. You shake it for a little while, and that detergent acts upon whatever cells it picks up and breaks those cells open, releasing ATP. When that ATP is in the fluid over there, it's available for the second important component within this tube. That's the luciferase enzyme. Luciferase is a light-producing enzyme. Very simply, it's very similar to the enzyme in fireflies, which you see them light up, or in seaweed when it glows in the dark on the top of the ocean. There are many of the similar enzymes present in nature. Those enzymes take ATP and make light. The more ATP, the more light. Very simple process. So by the enzyme being present in this tube, it uses the ATP present in that tube and produces light. That light can then be detected by this sensor here. So what is this method telling you? Well, it's telling you a certain amount of relative light units, or RLUs. We hear it used over and over again. That is relative to the dark in the chamber, how much light is produced. The more light is produced, the more ATP is present. The more ATP is present, the more cells were present, the dirtier the surface was. So in your facility, you would have a limit on which no more light than that should be detected from your surfaces. And then you know it's clean, very, very rapid. So it's a really quick, rapid method. It's used a lot in the food industry. It's used a lot in the hospital settings. It's not a substitute for plate counts and pathogen testing. Those are two things I'll discuss later on, but it's a very good supplement. It gives you quick, rapid results. Things to note though, you have to be careful as to what sort of surface you're sampling in. Different surfaces behave differently. Stainless steel is going to behave differently from wood, which is going to behave differently from ceramic, which is going to behave differently from plastics. And so you need a certain amount of knowledge of the surfaces and how they react. You also have to be careful of inhibitors. Because it's an enzyme in this tube, the enzymes can be inhibited by the chemicals that you use to clean. And so you don't want those chemicals to destroy the enzyme and change your results in the wrong way or make it too good or too bad. You want the real results that you see. And so generally I advise you just get an expert, spend a half an hour, one day talking about your system, and they will help you avoid the pain and hassle and design a proper system as part of your environmental program, which will enable you to use this really, really nice, simple tool and get good results as to how clean your processes are. What to do when it's too high a result, when you have too much ATP, the RLUs are too high, clean again. You can also take these results and compare them to your wider pathogen testing, your Listeria testing, your E. coli testing, your Salmonella testing. And you can have a huge system-wide verification where you have ATP data and also Listeria data and all the rest. And you can get a really good picture as to what is happening in your specific facility. Plus, the state government loves it, FDA loves it, all these regulatory agencies love that you're using such nice detection methods to keep your customers safe. So quick summary, ATP detection is used in food plants like yours. ATP detection is also used in hospitals at times, and both of those situations, it tells you how clean a surface is. Generally, the larger the number, the more dirty the surface, the smaller the number, the cleaner the surface. Design your system for your own environment. Your environment is going to be different from the next person's environment, so you have to be careful. And then there are lots of uses for ATP measurements, but we'll go to those in another video. We'll also talk a little bit more about what an environmental program and what it takes to do that in another video. For now, I want to thank you very much for watching. I want to thank our sponsors, Scientific Sanitation Solutions, Sanitsol. It's me, Dr. Ukpong. Once again, Dr. Yu. Comment, like, subscribe, and I'll see you all on the next video. Thank you very much.